This video is part of the Biopsychology playlist, which covers aspects such as the brain and its cells, neurophysiology, neurochemistry, neurotransmitters, and neurotransmitter dysfunction. I will discuss these now. Part 1. Neurophysiology. So to begin with, neurophysiology begins with the action potentials which carries information and neurons that communicate via the release of neurochemicals. In order to maintain a difference in the electrical charge between the inside and outside of cells, cells have a phospholipid layer, which is a plasma membrane that surrounds the neuron and allows for certain things to enter in and out of the cell. It allows for uncharged molecules to come in, and also allows in large amounts of potassium ions and few sodium ions to come in. Proteins and chlorine inside the cell helps to maintain net negative charge in the cell. There are large amounts of sodium ions outside of the cell and low amounts of potassium outside the cell. Ion channels allow for potassium ions to flow through the membrane and into the cell. There is also a sodium-potassium ion pump, which exchanges 3 potassium ions for 2 sodium ones. But in order to do this, it requires extra energy in the form of ATP, since it is going against the chemical concentration gradient. There are also voltage-gated ion channels that activate the opening of potassium when at minus or negative 50 to 30 microvolts, or when depolarization has taken place. All the opening and closing of channels allows for the creation of a resting membrane potential at around negative 70 MV, and when climbing up to the threshold of negative 55 MV, an action potential comes about and is an all or none, meaning that it causes an action potential to occur. This brings about depolarization, whereby more sodium rapidly enters into the cell by the peak of 30 MV. Repolarization occurs as sodium channels closes and potassium channels open in. Potassium ions are also of positive charge like the sodium, but sodium ions, or sodium Na2+, have twice the positive charge of potassium ions. Hence, a net loss of potassium charge culminates in growing negative charge. Then the charge reaches hyperpolarization, in which potassium channels still open for too long, causing the charge to dip below the resting state. Because of repolarization, a refractory period occurs in which the potential cannot travel backwards in the axons as channels are recovering. Also, there's a difference between absolute and relative refractory periods, in which absolutes cannot fire again while relatives will fire again if there is more positive charge. In addition, neurointegration is the culmination of information arriving to the cell from other neurons and occurs around the axon hillock. EPSPs, or Excitatory postsynaptic potentials, have positive charge and increase the likelihood for a charge to reach the action potential threshold. IPSPs, or inhibitory postsynaptic potentials, on the other hand, reduce the likelihood of reaching the threshold, thus inhibiting the neuron from starting an action potential. The synapse is the gap between the axon terminal of a postsynaptic neuron and the dendritic spine of a presynaptic neuron, which allows for the proliferation of neurotransmitters to facilitate cell communication. Different connection terms include axosomatic connections, whereby the axon synapse connects directly to the cell body; Axodendritic, in which the synapse connects to dendrites; and Axoexonic, where 2 neurons connect to the soma at the same time. Action potentials at the hillock causes an increase in calcium ions which then signals for vesicles to move to the membrane in order to dock and release neurotransmitters into the synaptic gap, which is known as the process of exocytosis. Neurotransmitters then bind to the receptors on the postsynaptic dendrites. Later, the neurotransmitters are unbinded and are transported back to the presynaptic neuron, known as the process of endocytosis, where they are either metabolized or recycled. Additional receptors to know are ionotropic receptors, which are also known as ligand-gated ion channels, that require ligand or chemicals to bind to the receptors in order for the chemical to open. These are composed of 5 subunits and are fast-acting but are ineffective since only one ion at a time enters through. Metabotropic receptors have G proteins that activate an ion channel directly or second messenger system. These receptors require a metabolic process to cause an effect and do not have subunits. Instead, they have 7 polypeptide spanning membrane domains, where each have alpha helixes where neurotransmitters can bind to. When G proteins are activated, the chemical GDP turns to GTP and is released across effector proteins or second messenger systems in order to activate many other ion channels. There are 2 types of G proteins. GS is stimulatory and GI is inhibitory. G protein coupled receptors are slow but have larger effect sizes, and it can activate multiple channels. There can be receptors on the presynaptic cell which provides feedback as to tell whether more neurotransmitters should be released or whether the releasing should cease. Other things to consider include agonists, which are drugs that activate receptors that work like neurotransmitters, and antagonists that bind to receptors but do not have an effect other than to block actual neurotransmitters from binding to the receptor. Therefore, in this video we looked at the neurophysiology of the cell, such as the phospholipid layer, ion channels, the NAK+ pump, voltage-gated ion channels. We also covered some neurochemistry like the action potential, depolarization, repolarization, hyperpolarization, and the refractory period. Neurointegration was looked at, including EPSPs and IPSPs. The synapses and types of connections like axosomatic, axodendritic, and Axoexonic were looked at. We also reviewed the process of exocytosis and endocytosis for neurotransmitters, ligand-gated ion channels, metabotropic receptors, agonists, and antagonists were also discussed. Thanks for watching.